2.5 billion years ago, a group of bacteria invented a way to split water molecules and harvest sunlight. They spread across the planet's oceans, pumping out a waste product they couldn't use. That waste was oxygen. For them, it was just garbage. For nearly everything else alive, it was the most lethal poison Earth had ever seen. The Earth before oxygen looked nothing like the world we know. The sky wasn't blue. Thick atmospheric methane scattered light into a hazy orange-pink, closer to the skies of Saturn's moon Titan than anything recognizable today. The oceans weren't blue either. They were a murky olive green, stained by dissolved iron leached from the seafloor over hundreds of millions of years. The air was unbreathable, not because it lacked oxygen, but because breathing it would kill you in other ways. Nitrogen dominated, mixed with carbon dioxide and methane. Volcanic gases added hydrogen sulfide to the cocktail. If you could somehow stand on an archean shoreline, the smell would hit you first. Rotten eggs, sulfur, the chemical reek of a world running on metabolisms we'd consider exotic. Life had existed on this planet for over 1 billion years by this point, maybe closer to 2 billion. It was everywhere, coating rocks in shallow seas, forming thick, slimy mats that trapped sediment and built up into structures called stromatolites. But it was entirely microbial. No plants, no animals, no fungi, just bacteria and archaea, single-celled organisms invisible to the naked eye, living in a planet-wide biofilm. These organisms ran on chemistries that have mostly vanished from Earth's surface. Methanogens, members of the archaea domain, consumed hydrogen and carbon dioxide and exhaled methane. They were phenomenally successful. Their metabolic waste product, methane, accumulated in the atmosphere, where it acted as a powerful greenhouse gas. This was critical because the sun at this time was 20 to 30% dimmer than it is now. Without that methane blanket trapping heat, the planet should have been frozen solid. Instead, it stayed warm enough for liquid water. Other microbes used sulfur compounds. They pulled energy from reactions involving hydrogen sulfide or sulfate, processes that released more sulfur back into the environment. Still others practiced a primitive form of photosynthesis, using sunlight, but not the way plants do. These anoxygenic photosynthesizers didn't split water. They used hydrogen sulfide, or dissolved iron, as their electron source, producing solid sulfur or rust as waste, never oxygen. This was the stable equilibrium of the Archean eon. A warm, dim, methane-wrapped world ruled by microbes that would die instantly if exposed to the air you're breathing right now. Somewhere around 2.7 billion years ago, possibly earlier, a lineage of bacteria stumbled onto a new trick. We call them cyanobacteria today, though blue-green algae is the common name, a misnomer since they're bacteria, not algae. What made them revolutionary was a single enzyme complex, photosystem II. Other photosynthesizers already existed. Purple sulfur bacteria, green sulfur bacteria, various other lineages. All of them used sunlight to drive chemical reactions that built sugars, but they were limited by their fuel source. Hydrogen sulfide, for instance, only exists in significant quantities near volcanic vents or in specific chemical environments. If you wanted to photosynthesize using hydrogen sulfide, you had to live near a vent. Cyanobacteria evolved a workaround. Photosystem II allowed them to crack open water molecules. Water is one of the most stable compounds in chemistry. The oxygen atom holds its electrons tightly. Splitting it requires an enormous input of energy, so much that the enzyme had to harness 2 photons of light simultaneously, using a cluster of manganese atoms as a catalyst. But once they'd evolved this machinery, cyanobacteria had access to an unlimited resource. Water was everywhere. Oceans, lakes, rivers, tidal pools, anywhere with sunlight and water, cyanobacteria could thrive. They were no longer tethered to vents or specific chemical niches. They could colonize the entire photic zone, every sunlit surface on the planet. The chemical equation was simple: 6 molecules of carbon dioxide, 6 molecules of water, energy from sunlight, and out came 1 sugar molecule and 6 molecules of oxygen, the sugar they kept. The oxygen they dumped. It was metabolic trash, a byproduct they had no use for. But for them, oxygen was just pollution, something to get rid of. They had no way of knowing they were about to terraform a planet. At first, the oxygen didn't accumulate. Every molecule cyanobacteria produced was immediately consumed by chemical reactions. The Archean oceans were a vast reservoir of reduced compounds, chemicals hungry for electrons, ready to react with any oxidant they encountered. The biggest sink was iron. Dissolved ferrous iron, leached from the seafloor by hydrothermal activity, saturated the water. When oxygen met iron, it oxidized it instantly. Ferrous iron became ferric iron, rust. And ferric iron doesn't stay dissolved. It precipitates. It falls out of the water as solid particles, drifting down like red snow to settle on the seafloor. This process lasted for hundreds of millions of years. As cyanobacteria spread, their oxygen output increased, but the iron kept absorbing it. The result is visible today in rock formations called banded iron formations, BIFs. These are some of the largest ore deposits on Earth, continent-spanning layers of alternating rust and silica, striped in rusty red and gray-black bands. Each band represents a cycle: oxygen production, iron oxidation, precipitation. The BIFs peak in the geological record just before 2.4 billion years ago, then taper off. The ocean was rusting. Slowly, over an incomprehensible span of time, the dissolved iron was being stripped out of the water, locked into rock. The color of the ocean itself was changing, from murky olive green to something clearer, eventually blue. But this was a buffer with a limit. Once the iron was gone, there would be nothing left to absorb the oxygen. It would saturate the water, then escape into the atmosphere. The sinks were filling up, and cyanobacteria kept multiplying. Around 2.4 billion years ago, the iron sink finally gave out. Oxygen began to accumulate in the oceans, then in the atmosphere. For the first time in Earth's history, free molecular oxygen, O2, was present in significant concentrations in the air. For the anaerobic organisms that dominated the planet, this was an apocalypse. Oxygen is not a passive gas. It's an oxidant, which means it aggressively steals electrons from other molecules. Inside a living cell, this creates what are called reactive oxygen species, free radicals. These are molecular fragments with unpaired electrons, wildly unstable, desperate to react with anything nearby. They rip through cell membranes, tearing apart the lipid molecules that hold the membrane together. They warp the structure of proteins, including the enzymes that carry out every chemical reaction a cell needs to survive. They attack DNA, breaking the strands, scrambling the genetic instructions. For an organism with no defenses, exposure to oxygen is cellular annihilation. It's not like being poisoned by a toxin that interferes with one specific pathway. It's systemic collapse, a cascade of destruction affecting every part of the cell at once. The anaerobic microbes that had thrived for billions of years had no such defenses. They had evolved in a world without oxygen. There was no evolutionary pressure to develop antioxidant enzymes, no reason to evolve repair mechanisms for oxidative damage. When oxygen arrived, they died. This was the Great Oxygenation Event's central extinction. We don't have fossils to count species losses. Microbial life doesn't fossilize well, but the geochemical evidence is clear. The dominant metabolisms of the archean, the methanogens and sulfur reducers, and anoxygenic photosynthesizers were either wiped out or forced into refugia. They retreated to the only places left without oxygen: deep ocean sediments, buried in mud where oxygen couldn't penetrate, hydrothermal vents, where the water chemistry was still anoxic, and eventually the guts of animals, where anaerobic bacteria still live today, protected from the oxygenated world outside. The planet's surface had been sterilized of its dominant inhabitants. Oxygen's effects weren't limited to direct toxicity. It also triggered a climate catastrophe. Remember the methanogens, the archaea that produced methane, the greenhouse gas keeping the planet warm despite a dimmer sun. Oxygen killed them. In the oxygenated surface waters where cyanobacteria thrived, methanogens couldn't survive. Their populations collapsed, but oxygen also destroyed the methane that was already in the atmosphere. Methane and oxygen react readily, especially in the presence of sunlight. The chemical equation is straightforward. 1 molecule of methane plus 2 molecules of oxygen yields carbon dioxide and water. Methane is a far more potent greenhouse gas than carbon dioxide. When oxygen converted atmospheric methane into carbon dioxide and water vapor, it effectively ripped away the planet's thermal blanket. The transition was geologically rapid. Within a few million years, atmospheric methane concentrations plummeted. Global temperatures followed. Ice began to form at the poles, then spread. Glaciers advanced, growing thicker, reflecting more sunlight back into space, which cooled the planet further, a runaway feedback loop. This was the Huronian glaciation, named after rock formations in the Lake Huron region of North America, where the glacial deposits were first identified. It began around 2.4 billion years ago and lasted, with interruptions, for as long as 300 million years. Some models suggest the ice sheets covered the entire planet, a Snowball Earth scenario where even the equatorial oceans froze over, leaving only a few pockets of liquid water near volcanic hotspots. Life clung to those pockets. It nearly went extinct. The cyanobacteria that had triggered the catastrophe survived, ironically, because they could still photosynthesize under ice if enough light filtered through. But the diversity and abundance of life collapsed. The planet became a frozen wasteland, colder than any ice age humans have ever known, and it stayed that way for longer than the entire history of complex animals on Earth. Evolution doesn't stop during an extinction, it accelerates. The organisms that survived the initial oxygenation weren't the strongest or the fastest, they were the lucky ones, the lineages that happened to carry mutations that provided even marginal protection against oxygen's destruction. The first line of defense was antioxidant enzymes. Proteins like superoxide dismutase could capture free radicals before they caused damage, neutralizing them through controlled chemical reactions. These enzymes didn't prevent oxygen from entering the cell. They couldn't. But they could intercept the reactive oxygen species, the molecular shrapnel, and convert them into less dangerous compounds before they shredded membranes or fragmented DNA. Organisms with these enzymes could tolerate oxygen. They weren't thriving yet, but they weren't dying. That was enough. But tolerance wasn't the end of the story. It was barely the beginning. A subset of survivors went further. They evolved a new metabolic pathway, one that didn't just tolerate oxygen, it weaponized it. This was aerobic respiration, and it required rethinking the entire energy economy of the cell. Anaerobic metabolisms, like fermentation, had dominated life for billions of years. They worked through a short series of chemical reactions. A glucose molecule, a 6-carbon sugar, would be broken down into smaller fragments. In the process, a small number of electrons would be transferred to carrier molecules, which would generate a modest amount of ATP, the cell's energy currency. The yield was about 2 molecules of ATP per glucose molecule. It was inefficient, but in a world without oxygen, it was the best option available. There was no alternative electron acceptor powerful enough to extract more energy from the fuel. Oxygen changed the equation. Oxygen is an electron thief, and a greedy one. It pulls electrons with more force than almost any other molecule in biology. Early aerobic organisms evolved a way to exploit that pull. They built a molecular assembly line, a chain of proteins embedded in the cell membrane, each one passing electrons from one to the next like a bucket brigade. At the end of the line sat oxygen, waiting. When the electrons finally reached it, oxygen grabbed them, combining with hydrogen ions to form water. That final transfer released a burst of energy, far more than any anaerobic pathway could generate. The system was called the electron transport chain. It didn't just break down glucose more completely, it extracted every available electron from the molecule, wringing out energy at multiple steps along the way. But the most consequential event came roughly 600 million years after the Great Oxygenation Event itself, around 1.8 billion years ago. This is when one lineage of bacteria that had mastered aerobic respiration entered into a partnership that would reshape the trajectory of life. The details are hazy. This is deep time, and we're reconstructing events from genetic and biochemical clues. But the broad strokes are clear. A larger cell, possibly an archean or an early proto-eukaryote, engulfed a smaller aerobic bacterium. Normally, the bacterium would have been digested, broken down for nutrients. But something went wrong, or went right, depending on your perspective. The bacterium survived inside the larger cell. It kept respiring. It kept producing ATP. And the host cell kept it alive, feeding it, protecting it, because the energy surplus was too valuable to destroy. This wasn't a one-time event. The partnership had to stabilize over generations. The bacterium had to lose its independence, shedding genes it no longer needed. Its host provided nutrients, so why maintain the genes for hunting or building a cell wall? The host had to become dependent, outsourcing energy production to its captive. Over time, the bacterium became an organelle. We call it the mitochondrion. Every eukaryotic cell on Earth, every cell in every plant, fungus, animal, and protist contains mitochondria. They still have their own DNA, a stripped-down remnant of their bacterial genome. They still have a double membrane, evidence of that ancient engulfment. They reproduce independently inside the cell, dividing when they need to, just like free-living bacteria. They are living fossils, descendants of the microbes that survived the Great Oxygenation Event by learning to harness the poison. Without mitochondria, there would be no eukaryotes. Without eukaryotes, there would be no multicellularity. No cooperation between specialized cells, no division of labor, no tissues or organs. Multicellular life is energy expensive. Building a body with trillions of cooperating cells, some of which do nothing but transmit signals or contract or secrete chemicals, requires an energy budget that anaerobic metabolism simply cannot provide. The mitochondrion made that budget possible. This is why we exist. Not because oxygen is inherently good or because aerobic respiration was inevitable. We exist because a few lineages, through accident and selection, turned a global poison into a power source. The descendants of those lineages inherited a planet that had been scrubbed clean of competition. They inherited an atmosphere rich in the very molecule their engines required. They didn't survive the apocalypse despite the oxygen. They survived because of it. Cyanobacteria didn't plan any of this. They were bacteria. They had no foresight, no goals. They evolved a useful enzyme. It gave them a competitive advantage, and they multiplied. The oxygen was incidental, waste, nothing more. But that waste restructured the planet. The sky turned blue. Oxygen molecules in the upper atmosphere, bombarded by solar radiation, rearranged into ozone, O3. Ozone absorbs ultraviolet light. Before the ozone layer formed, UV radiation sterilized Earth's surface. Life had to stay underwater, where the water itself provided a shield. Once ozone built up, around 600 million years ago, the surface became habitable. Plants could colonize the land. Animals could follow. The oceans turned blue. With the dissolved iron gone, the water cleared. Photosynthesis, now performed by cyanobacteria, algae, and eventually plants, continued to churn out oxygen, maintaining atmospheric concentrations around 21%, the level we live in today. The climate stabilized, though it never returned to the Archean's methane-warmed constancy. Earth entered a new phase, cycling between icehouse and greenhouse states, driven by shifting levels of carbon dioxide, continental drift, and the biosphere's own activity. But it was a dynamic equilibrium, not the static methane blanket of the past. The energy economy of life had fundamentally restructured. With aerobic respiration available, complexity exploded. Eukaryotic cells emerged. Multicellularity evolved multiple times. Animals appeared, then diversified in the Cambrian explosion 540 million years ago. a geological eye blink compared to the billions of years it took to get there, but unthinkable without the oxygen that aerobic respiration requires. The cyanobacteria poisoned a world. They triggered the largest extinction in Earth's history. They caused a climate collapse that nearly ended all life. And in doing so, they created the preconditions for everything that followed: forests, reefs, dinosaurs, humans. The apocalypse they unleashed was also the foundation for the only version of Earth that could produce organisms capable of understanding what happened. We live in the wreckage of an accident. Every breath you take is made possible by an ancient catastrophe, a mass extinction that cleared the planet of its original inhabitants and replaced them with a new species, them with lineages that learned to harness the poison. The oxygen in your lungs is the same waste product that killed nearly everything alive 2.5 billion years ago. We're not survivors of the Great Oxygenation Event, we're its beneficiaries. The world cyanobacteria destroyed is the only reason we're here to tell the story.